Welcome to the Outperform Cancer podcast, where we identify anti-cancer strategies found in peer-reviewed scientific research. My name is Mary Beth Gilliam, and I'm a metastatic breast cancer patient searching for ways to outperform cancer. I am very grateful for the excellent standard care I've been given, but I'm also not satisfied with standard care results because I think it's possible for cancer patients to do better. And I believe the people who can help us get there are cancer researchers. So, in each episode of our podcast, we talk to a well-regarded published researcher who's focused their work on a potential anti-cancer strategy that could be used with conventional treatment. Visit the Outperform Cancer website at outperformcancer.com to see all of our anti-cancer strategies and to sign up for our newsletter, which will alert you to new strategies as they're posted. Today, we're speaking with Professor Dr. Connie Jimenez about high-dose vitamin C. Dr. Jimenez is a professor of Translational OncoProteomics and the principal investigator of the OncoProteomics Laboratory at Amsterdam University Medical Center in the Netherlands. She's also the vice president of the European Proteomics Association and co-chair of the Human Proteome Organization, as well as winner of their Translational Proteome Science Award. She's authored more than 200 peer-reviewed scientific papers and is an editorial board member of 5 major proteomic journals. Wow, yes. She is an incredibly accomplished scientist, and her brain power is about to help us because Dr. Jimenez's work involves an anti-cancer strategy for us, and it's called high-dose intravenous vitamin C. In addition to her laboratory work, where she tested the effect of high-dose vitamin C on 51 different cancer cell lines, she also led and published a meta-analysis in 2001 titled High-Dose Intravenous Vitamin C, a Promising Multi-Targeting Agent in the Treatment of Cancer. This paper could be my all-time favorite because not only does it include 71 preclinical studies, 57 clinical trials, and 24 omics studies in one analysis, but it makes the information easily accessible to cancer patients. So easy, in fact, that there is a color-coded chart organized by standard cancer drugs that allows you to look up your drug and see the results of the intravenous vitamin C research when combined with your drug. I looked up the drug trastuzumab because that's part of my standard care therapy. And I found that there's an IVC, intravenous vitamin C, study that was conducted specifically with trastuzumab. The study found that IVC made trastuzumab more effective. I think that's critical knowledge to have. Now, without further ado, we're going to talk to Dr. Connie Jimenez, who will explain IVC and why it's such a powerful anti-cancer strategy. Thanks. Thank you for coming today, Dr. Jimenez. We really appreciate your time.
Thanks, Mary Beth. It's my pleasure to be here on your podcast.
I'm hoping that you can start us off today just by talking a little bit about your work. Can you talk a little bit about that?
Oh, yeah, sure. Thanks for the opportunity. So proteomics, it's a research field that focuses on the large-scale unbiased analysis of protein profiles in cells, tissues, body fluids. And OncoProteomics is the application of proteomics to study cancer. And this is very relevant for patients with cancers because proteins carry out the cellular functions. Proteins determine which pathways are activated in a patient tumor. And for example, whether a tumor is aggressive or benign, whether it spreads or not. And what is more well known is that our genome or DNA, it's heavily aberrant in patient tumors. And that informs on what can happen or predisposition, while the proteome informs on the actual signaling state of a cellular cell or tissue.
Now, let me just ask you a quick question because I've been reading a lot about the expression of our genome, and what I've been hearing is that things can get turned on and they can get turned off. Is that true? And is that part of your work to see what's turned on and turned off?
Yeah, exactly. Things get turned on and turned off, and it's kind of hard. There is not a one-to-one relationship from DNA to the messenger molecules, the RNA and protein. So based on DNA alone, it's hard to know what's happening or predict what's happening, and this is where the proteome comes in. And maybe also good to realize that most biomarkers that are measured in the clinic are proteins. And proteins are the targets of most anti-cancer targeted therapies, such as kinase inhibitors and immune checkpoint inhibitors. So in our OncoProteomics research, we study these protein profiles in cancer cells or tissues. And we hope to use this information, we couple it to all kinds of bioinformatic analyses to better understand cancer biology and employ this information to drive improved diagnostics and individualized treatment of patients with cancer. And
Pardon
yes.
me. That sounds like kind of where we're heading in terms of more personalized treatment for patients. You mentioned targeted therapies, which is that first step of not the widespread traditional chemo, which makes us all so terribly sick as it tries to wipe out the cancer. So the targeted therapies, but also kind of the next level, perhaps, of really understanding how each one of us is how the tumor for our individual tumors are expressing itself and how it's growing, its pathways that it's using for growth. Is that part of this too? Is this moving us closer to more personalized cancer treatments?
Well, yeah, I hope so. That's what we're aiming for, and this is really important because what we see in our own molecular profiling efforts, and also what can be seen from the DNA and the RNA levels, is that basically every patient tumor is different. Even though you may have a whole set of colorectal cancer tumor profiles, but in the end, if you take multiple samples of the same tumor, they cluster together, and every profile is different, has different aberrations, different activation of the pathways, different combinations of parallel activities. And so it's very important to basically also have this functional layer of information that we can get from the proteome and the phosphoproteome. And to do this, I have a whole multidisciplinary team of people, and not only people with biomedical biology background, but also biochemists, computer science, and other data analysis people. So it's a whole clinicians, of course, to give us the clinical background and to make sure we work on the clinical needs. It's a whole effort to do this.
That's amazing because it's actually something that I pay a lot of attention to because I believe when you're working in a multidisciplinary team, that's what's required to help us move from a preclinical discovery, something that was maybe found in a lab in a microenvironment, and to move it into a clinical trial, which gets us closer to making it part of standard care. And right now that process takes about 15 years, which unfortunately, for most cancer patients, it's just too long. We're not going to be seeing the benefits of those discoveries. So working in multidisciplinary teams is really exciting because it means that distance in knowledge from your work, which is happening on a micro level, to the bigger work in humans that happens, that you're all in the same room. Is that true? Is that what I'm hearing?
Yes, indeed, that's what you're hearing, because it takes a huge effort to translate findings from the lab to the clinic, and it really has to be done in very close collaboration with the clinicians. And it will only happen if if we work in the lab really on clinical needs, on the right materials of the right quality, with good clinical and molecular annotation. So yeah, it's very important. So I think the embedding of my laboratory in a academic hospital, in the Department of Medical Oncology, I think is very important to, let's say, to increase chances of making these translational steps. And of yeah,
That's
that's
awesome. It's really encouraging to hear. And I have to say that, I reached out to you because the research that you did, a paper that you authored, titled "High Dose Intravenous Vitamin C: A Promising Multi-Targeting Agent in the Treatment of Cancer," it's really my favorite study, and I'll tell you, I just found it to be very insightful and clear. And I know I'm not the only one because this research paper has actually been cited in about 80 other research papers in barely 2 years. So that's a lot of connectivity within the research community, which indicates that it's pretty great piece of work. So can you just tell us what led you to investigate IVC, high dose IVC, intravenous vitamin C, and why did you embark on writing this meta analysis, with your team?
Yeah, well, my fascination for vitamin C was ignited about 7 years ago, I think, when I discovered in my personal life that vitamin D and C in high dose can have therapeutic effects. And then I started doing more reading on it, and I think it's good to realize that vitamin C is an ancient and essential molecule of life with widespread biological functions. Its appearance during evolutionary times was probably very important for the development of life on Earth. And all cells in our body take up vitamin C. They need vitamin C to function. It's the most important antioxidant in our biofluids in blood. And antioxidants are very important because they can neutralize the damage done by free radicals. And then vitamin C has a whole lot of other functions that have been discovered in more recent years as cofactor of many enzymes that get the electron from vitamin C to drive their activity. And that can explain why it can influence so many biological processes in our body.
So that sounds like kind of a essential underpinning, if you will, for health overall, which of course we know for cancer patients is amplified because our body is under increased stress, both from the cancer as well as from the treatments, from many treatments.
Yes, indeed, indeed. For example, vitamin C is very important for the synthesis and the structure of strong collagen. And of course, that's a very important connective tissue structure that can help us or can protect patients from cancer against spreads. Uh, it's um, it's involved in the synthesis of neurotransmitters, in steroid hormones, stress hormones, energy production, cholesterol metabolism, detoxification, iron absorption, and much more. So key for the healthy functioning of the body, but also, of course, maybe even more key functions to help patients with cancer battle their disease.
Yeah, and it's funny, the things that you mentioned, when I first was diagnosed, I didn't realize the connection between cancer and many of those things. But now I understand better, and we'll talk a little more later about how it is connected to iron absorption and processing of lipids and because all of those things actually contribute to whether the disease whether making an environment that's easy for the cancer to metastasize and spread or whether we're making it more difficult. So it sounds like this is a important underpinning.
Yes, and then I think what I did not mention yet is the important effects of vitamin C on the immune system, our bodily defense system. And to underscore this important function, our immune cells have the highest concentrations of vitamin C in our body. So it's very crucial for immune function, and other very high concentrations are in our brain, in the pituitary, and also in the adrenals, for example. So very important in the also to support our stress or system to handle stress and stress hormones.
That
So yeah, and I think it's such a basically from the evolutionary perspective an important molecule, and humans lost it during evolution or sometime during the primates evolution, because we cannot synthesize it in our bodies. Monkeys cannot synthesize it, and humans cannot synthesize it. And basically, the rest of the animal world and the plant world, they make their own vitamin C, and they make more when they need it, when they're sick or under stress or injured. So basically, the vitamin C word is kind of confusing, because actually it's more like a hormone, should be seen as like a multifunctional hormone, and also stress hormone. So that kind of implies that yeah, we need a lot of it, and we should look at for how much we need. We should look at what other animals make in their bodies.
that's okay. I've never heard that before. It's fascinating. So let me just sum this up. So what you're saying is that, at one point in time, probably all life synthesized their own. That means created, their bodies had the ability to create their own vitamin C. But somewhere along the line, primates and then humans lost that ability. And so now the only way we can get vitamin C is to artificially get it. So ingest it through our food and what we eat, or through intravenous vitamin C. But somehow we have to get it externally, whereas pretty much all the other animals and plants out there can make it themselves. Like they don't have that problem. There's that big of a difference between humans
Yeah,
and
indeed.
everybody else.
Yeah, I'm a little bit jealous on animals. Yeah, no, yeah, we lost so basically the last so is vitamin C is synthesized from glucose in a couple of enzymatic steps, and in the last enzyme is not functional in humans and monkeys, and also in guinea pigs. So they are a nice model. And then there's a fruit bat. Basically, these types of animals, but the vast majority can synthesize it, and the warm-blooded animals synthesize it in their liver. Cold-blooded animals synthesize it in their kidney, and basically also in throughout during evolution, when from cold-blooded to warm-blooded, it moved the synthesis primary spot moved to the liver. Probably the liver is much bigger, can accommodate much more to support the body with higher levels of vitamin C.
So I always think about it. I think most people, think, oh, vitamin C, you take it when you're sick. But I don't know if they are associating it with stress. So it's actually necessary, or works with our bodies, helps our bodies under times of stress. So that could be not just I have a cold, but any kind of stress, right?
Yeah, and any kind of stress, and to for the production of stress hormones, you need vitamin C. So if you have a lot of stress, you will, by definition, need more vitamin C. So in animals, that can synthesize their own, it's dynamically regulated. But we in our bodies, it's absent. So we have to take it through our diet. So either eat lots of vegetables and fruit, fresh as fresh. Otherwise, it's lost. It's heat sensitive. Or supplement, and to get in the gram range, supplementation is the solution in let's say for the modern day humans.
Wow. And so in an average person, you said in the gram range. About how many grams for just a normal, healthy person
Yeah.
be looking at, which to combat a lot of this stress? And can we take it at one time, or do we need to divide it in the day?
Well, it's probably a good idea to supplement 2 to 3 grams in divided doses over the day, because what the body cannot take up, it's secreted through the kidneys, excreted. So we have to continuously have a supply, and that is best done by supplementing 2 or 3 times or more, depending. If you're sick, it's a good idea to do more.
Mm-hmm. Let's think about cancer patients specifically. Going back to your research paper that you did, you talk about high dose vitamin C. Can you tell us a little bit about the difference between what we're talking about with a relatively healthy person taking some little extra vitamin C, or getting it through a little extra fresh fruits and vegetables, versus the cancer patient that might need a different, more intense strategy if you want it to be an anti-cancer strategy?
Yeah, right. So what the review is about, that's about high dose vitamin C. So that's really therapeutic use of vitamin C. And that's a whole different ballpark because that needs to be given intravenously in like mega doses. So we're talking here at least 1 gram a kilogram. So a 70-kilogram person would get 70 grams intravenous. And this was kind of pioneered in the 70s by Linus Pauling. So this is when I started reading on vitamin C, I stumbled upon this literature and the documentation of also there was clinical experience. And so this is like a whole different. So it's very important to discriminate oral use of vitamin C. Then that can only reach up to in blood in the 220 micromolar range, versus intravenous vitamin C that can go up into the 15, 20, 30 millimolar range. And that's the range that you need to kill cancer cells, that we know from in vitro studies.
So just to be clear, if we want to make this an anti-cancer strategy, maybe to augment standard care that we're getting, if I wanted to have a high dose of vitamin C, I could not go take a whole bunch of or orally take a whole bunch of vitamin C tablets, and it would not work, right? It is a very different concept. It acts very differently in your body to take, an extra vitamin C tablet in the morning with breakfast, versus even if you were to take a whole bunch of those, it's very different than vitamin C that you get intravenously, right?
Yes, because if you take it orally, at some point the uptake mechanisms in the gut get overwhelmed, and so there's only so much that they can take up, and then the kidney starts excreting it. So that's why with oral use we cannot go higher. It's tightly regulated. And with the intravenous administration, we can bypass these control mechanisms.
Okay, so for cancer patients, then, just to be clear, we're talking about high dose vitamin C that's done intravenously as a potential anti-cancer strategy, and then moving forward in this discussion, that's really what we're going to focus on in the next bit of our conversation. So, because there is a big transition there, and I believe taking it orally is a antioxidant reaction in your body, versus through intravenous vitamin C, which is pro-oxidant. Is that true? So our bodies even process it differently?
Well, for the cancer cells its pro-oxidants in these high doses, whereas for the rest of the body, it can benefit from the antioxidant effects. And that has to do with the biology of the cancer cell, why it's a pro-oxidant. And I think we'll go into that in a minute later.
So you wrote this amazing research, which I loved. And one of the reasons I liked it so much is that it dovetailed with what I kind of believe in my gut to be true, which is that layering different strategies on can help cancer patients do better in the long run. In fact, in the 1960s, this idea isn't new. In the 60s, oncologists discovered that when they combined chemotherapies, patients had better outcomes. They started putting 2 together, 3 together, 40. And now patients might get a cocktail of 6 or 7 different chemotherapies, all to combat a particular cancer that they have. But this combined effect, because cancers grow through so many different pathways, this combined effect can be more powerful, can shut it down on multiple levels. So your research was so interesting to me because your summary in particular showed intravenous vitamin C when used in conjunction with standard care. You have this terrific chart that uses symbols and colors to indicate the impact of IVC when used with standard care treatments. So I could look up my drug in particular, trastuzumab, and see that there was an in vitro study showing enhanced efficacy for the drug I'm on, trastuzumab, when combined with IVC. So in total, cancer patients out there, take note, in total, there were 59 different drugs or treatments in your study that you addressed. So cancer patients can actually go to this chart, and we will put it in the show notes, a link to this research, so that they can look up their drug that they're on and see what research has been done with IVC and their drug. I thought that was fascinating. So can you tell me, prior to this analysis that you did, were you aware that IVC had been tested with so many different cancer drugs? I was amazed. Are there any particular findings of efficacy or synergy that really stood out to you? I mean, is it just me that got super excited about this, or what was your impression when you started to write this report?
Well, yeah, I was actually quite amazed about the work out there, what was done, and when I started reading that, I thought this is almost too good to be true. And all these anti-cancer effects and all these combinations that were already tested, and most of them synergistic. So yeah, when I kind of discovered this whole literature, then 5 years ago, I put it all already some years ago. I put it together in a PowerPoint, and when it was my turn for the department staff meeting, brainstorm evening, I presented this work on vitamin C or the high dose in the cancer context to kind of let my oncology colleagues know and share the excitement, and did they know this? No, they actually did not know, and they were actually most of them were actually quite interested, intrigued. So we had some great discussions. And since this was also new information for them, this is what really made me decide to write all this data out there up in a nice review. Do our best, I should say. I did it together with the help of together with PhD student Andrea Valles, postdoc Francesca Butcher, and also a master student who did her literature thesis on this topic. So we really did our best to make comprehensive figures, to summarize all the relevant details in the tables, to make it really easy for oncologists. I mainly had the oncologists in mind as a lookup thing. and also to make them aware of the wealth of information, and that it can really be safely combined with basically all the therapies out there that they're using. And so I'm very happy to hear from you that also it works for you. And now I really want to, I also decided to start my own research line in the lab with high dose C, testing it on cancer cells to see for myself if this really works. And so it really brought a whole range of new things.
That's amazing. Yes, and I really encourage cancer patients to go to this study, print this chart out, and bring it into your oncologist, because I know my oncologist was not aware, and it's a simple chart that summarizes all the research, and you just look up your drug. In fact, cisplatin, for example, is one drug that I know many cancer patients, many different types of cancers, are treated with this particular chemotherapy. And cisplatin had a number of different studies that had been done with high dose vitamin C.
Yeah, I think a lot of studies made use of platinum-based chemotherapies, and I think there, of course, there's a lot of toxicity related to that. So I think there the need to also counterbalance this toxicity with vitamin C is highly desired. So I think it was great to see that all these preclinical studies showing synergy, but also that there is several clinical phase 1, phase 2 studies done showing the safety and the feasibility also in the clinic. So and then the cell line studies show synergy. The mouse studies also show that it can really reduce the tox from the chemo. It decreases white blood cell loss, weight loss, ascites formation, hepatotoxicity, toxicity of the liver, lipid oxidation, and other things like cardiomyopathy. So in the mouse, it's clearly beneficial. And then in from the clinical studies, or actually one study that goes from cell line to mouse to phase 1 to clinical trial, is the study that was published in Cancer Cell a few years back on glioblastoma and non-small cell lung cancer. And the non-small cell lung cancer part of the study, there vitamin C high dose intravenous was combined with the platinum-based treatment. And also in the cell line studies, the cisplatin was used. And it really showed synergizing effects in the cell lines. And then the trial, which of course is only early stage trial, at least it underscored that vitamin C or pharmacological doses are safe and well tolerated when combined with this standard therapy in both glioblastoma and non-small cell lung cancer. And this study was not powered for efficacy. Yet what the authors write in the paper is that overall survival was better than historic data for comparable populations, and that of the glioblastoma patients, even a few 4 out of 11 remained alive at the time of writing, and one of them even didn't show any evidence of disease anymore as based on MRI. So these are very encouraging results. And there are several clinical trials, also randomized some, that show similar results.
Wow. Okay, so there's two things that I just wanted to capture at this moment. First, going back to what you were saying earlier, was that the high dose vitamin C was shown to help with some of the issues around cancer patients' treatment. So, putting aside for a moment trying to kill the cancer cells, the fact that the medications that so many of us are on are so toxic, and we suffer from things like cardiomyopathy, I think it's important to point out what you were saying earlier, because many cancer patients can actually die because of the cancer treatments. So for example, I'm on a drug called trastuzumab, and it has shown that it can create problems with the heart, right? And so I have to have an echocardiogram every 3 months to monitor my heart. So the first point you were making that I want cancer patients to take note of is that high dose vitamin C has been shown to mitigate those negative other effects.
Yes, and the high dose intravenous, of course, is a great fast way to get it. But I think in the days, these administrations usually are only 2 or 3 times a week. So I think in the in-between days, to help with all these other side effects of chemo, I think the high dose oral supplementation could also be already beneficial.
Okay.
But for sure, the intravenous administration helps with that a lot. But the intravenous part is especially important to get the cytotoxicity for the cancer cell.
Right. So that gets to the second thing that you were saying. So the high dose vitamin C is will give you this other effects to help with toxicity and quality of life issues and cardiomyopathy and things like that. So it helps those things, but also helps with actually killing cancer cells. And that's the research study that you were mentioning about glioblastoma and non-small cell lung cancer, that research study from 2017, which again, we'll put into the show notes so that people can look this up for themselves. And that was the second thing I wanted to highlight, because that's exactly why we're doing this podcast. Here we have some research that has been taken from the preclinical level. So really understanding what happens in a petri dish, what happens in a mouse model, and then translating that into human clinical trials, phase 1 and phase 2, where we get very good results across the board of that research. So in the different types of methodologies, we're seeing consistently positive results. And for eliminating cancer cells, for moving cancer patients in the right direction. And this has been layered on top of in the phase 2 and phase 1 clinical trials, layered on top of standard care. Here's something that's positively happening all the way through phase 2, but unfortunately, we don't see phase 3 trials. And without a phase 3 trial, it's never ever going to be part of standard care, and where we can walk in and expect this our doctor or oncologist to be giving this to us. You know, we've gotten to this point of phase 1, phase 2, but so much of this research stalls. And then people don't hear about it and they don't know about it.
Yes, and I would say there is huge biological rationale, because we know why it works and how it works for at least for the most well-known mechanisms, and a lot of details were also unraveled in this cancer cell study. So it was known already for a bit longer that in these high doses, vitamin C works as a pro-oxidant, not as an antioxidant, but as a pro-oxidant. And it results in high levels of hydrogen peroxide in the cancer cells that are really selectively toxic to the cancer cells. And then a lot of details or there's a good understanding why this is, because these cancer cells, they are really basically messed up weird cells. They have a redox imbalance, which means that there's a lot of oxidative stress in the cancer cells due to their increased metabolism, among others. They have higher levels of iron in them, or the what's called the label iron pool, and vitamin C in high dose interacts with that. The cancer cells have defective mitochondria causing this oxidative stress. They have the decreased antioxidant defense system. So that's why it's really selectively toxic to cancer cells, and then and normal cells, they are not bothered because they have antioxidant system in place, so that they only get the benefits, not the pro-oxidant effects. And then also some studies have shown, or we know that cancer cells, they to support their glycolytic metabolism, they need a lot of glucose. So they have upregulated the glucose transporter on the cell surface of their cells. And what is so interesting is that vitamin C, it's synthesized from glucose. It has a very similar structure to glucose. So vitamin C in oxidized form especially can be taken up by the glutamate transporters on the cancer cells. And then that's like a Trojan, works like a Trojan horse, because then oxidized vitamin C is taken up, and then inside the cell it's reduced again, and this further depletes the antioxidant defense of the cancer cells. So it's really a very nice targeting effects. And highly synergistic with chemo and radiotherapy that also act as pro-oxidant. So it can help drive the cancer cells over the cliff, while the normal cells and the immune system benefits. And this is especially important because cancer cells are very oftentimes really low in their vitamin C. So there are some of them are at scurvy levels. So with this can really help their body a lot. And then there is more like secondary effects, you could call them, how vitamin C works in high dose to beat cancer, and that is more in the cofactor activity of vitamin C. So that I already mentioned it. So it helps support collagen or connective tissue structures in our body that can help impede cancer spread. It can, very important mechanism is also it can inhibit hypoxia-inducible factor activity. And then HIV is broken down, and HIV is associated with more aggressive disease, and it's important to get blood supply to the fast-growing tumor. So it's a way to cut off the fuel. It can also help, these were studies in pancreatic cancer, reverse epithelial mesenchymal transition. That's a process that's also implicated in cancer metastasis and poor prognosis, can help reverse that. And then another more recent discovered mechanism also is the reversal of aberrant DNA methylation by through its cofactor activity for the TET enzymes, which results in reactivation of tumor suppressor genes that can help keep the cancer in check and reinstate apoptosis. And then, of course, last but not least, it can help boost the immune response. And I think for this, it's also to basically help the host attack the cancer. It's vitamin C has widespread functions in the immune system. It helps with myeloid and T cell differentiation, polarization, T cell maturation, activation, B cell development, cytokine production, and it can enhance the natural killer cells that are very important to attack cancer cells.
Wow. Okay. So what I heard was that it's sneaky. So it can be the Trojan horse. I love that analogy. The Trojan horse to get into tumor cells, into cancer cells, because of its similarity to the glucose molecule. So it's sneaky. It gets in. It can cut off the fuel for the cancer cell, so making it harder to grow, metastasize, progress, and harder to even stay alive. So it can kill a cancer cell that way. It provides a reversal of what we see where a tumor suppressor gene might get turned off, meaning if it was suppressing tumors, it wasn't letting the tumors grow, that tumor suppressor gene, when we get cancer, sometimes is turned off. And now there's no suppressor. So the tumor can grow unchecked. But the vitamin C, high dose vitamin C, can reactivate that tumor suppressor gene. And that also allows it to help kill the cancer cell and avoid metastasis. And then on top of that, we also see a boost or an elevation of immune response. So T cells, like the killer, natural killer cells, we've talked about T cells and B cells. So we can boost those. And we know now so many therapies for cancer patients that have a lot of promise are immunotherapies. So are we thinking that this would work synergistically also with immunotherapy? That comes to my mind.
Yes, so when I was because of these very important immune functions, one could hypothesize that the combination of high dose vitamin C together with immunotherapies, checkpoint inhibitors, is a good idea.
Mm-hmm.
And I hypothesized that also a couple of years ago. And then like 2 years ago, there were two preclinical studies in the mouse where exactly this combination was tested, and also there it's really beneficial to combine high dose C with immunotherapy. And what was also interesting is that the note that the authors made is that it's very important to do this in an immune competent mouse, because in a lot of the studies use these immune deficient mice, but you get much more of the high dose vitamin C effect in immune competent mouse, and that kind of confirms the idea that the effects of high dose C are both through their direct effects on the tumor, but also the more indirect effects via the immune stimulatory function.
Mm-hmm.
Yeah, they're all immune checkpoint inhibitors. Yeah.
Okay. So
the ones tested were anti-PD-1 and anti-CTLA4.
okay. Okay, good. Because I think, it's nice is for some cancer patients, I'm hoping that people will see themselves or hear something in these podcasts that said, hey, that's me. I can relate what you're saying to my care. And so I think it's nice to be able to point out to them exactly what checkpoint inhibitors, because that might be something that's on their list. And that gets us to what I'm hoping you'll talk about next, which is certain mutations that seem to have a lot of reactivity with a high dose vitamin C. So, in other words, there's been a lot of promise in research with certain tumor mutations. Can you speak a little bit to that?
Well, both the preclinical and the clinical research shows that patients actually with a large variety of genetic aberrations may benefit from the combination treatment of high dose C with standard of care therapies. And what is encouraging to see that this also includes hard to treat patients with, for example, KRAS mutations. KRAS mutations are found in 90% of the pancreatic cancers, 40% of the colon cancers, 25% or so in the non-small cell lung cancer. So, and that's a mutation that gives poor prognosis, and so even those patients can benefit. A lot of research has focused on pancreatic cancers, where most of them are KRAS mutant. And there's also there's a clinical trial that shows that these patients had improved performance status. Their mean time to progression was much longer than what would have been expected, again from comparable historic populations. And this was then in the context of gemcitabine. So there was a significant improvement compared to trials that used gemcitabine alone. and this was and there's another trial that was published in Cancer Research in 2018, also in pancreatic cancer. Nice randomized study that showed the same results. So a lot of promising, let's say, very early stage studies, also in metastatic colorectal cancer, where actually that study reported comparable efficacy in patients with wild type and mutant ROS and BRAF. And besides these authors, besides a favorable safety profile, they also reported potential clinical efficacy with a disease control rate of in total 96%, combining partial response and stable disease in the population. So based on these promising results, actually they are planning a phase 3 study.
Oh, that's
which is
exciting.
highly needed, yes.
Yes, it is highly needed. So that study that you're saying that will move to phase 3 is with KRAS or BRAF mutations. Is that what you're saying?
I think in that study they include all wild type and mutant, but I know that there is a study in the US from the Stand Up to Cancer effort. They are specifically focusing on the KRAS BRAF mutant setting.
Okay.
All in right. Metastatic colorectal cancer.
Okay. We will make sure in the show notes to also post clinical trials that are associated with some of the things that we're talking about. So cancer patients look up and see whether they might be someone that would be a good fit for some of these trials that are underway or coming up soon. Can you tell me what about breast cancer and prostate cancer? I haven't heard very much about those two cancers in particular, and they affect so many people. Would IVC be a potentially beneficial strategy for those two cancers?
Yes, sure. I don't think that there's a reason why they shouldn't be. I think the studies out there probably are focused a little bit more on the hard to treat cancers, and that's maybe a reason why they're underrepresented. At least from our own results in the cancer cell line panel, we had a couple of breast cancer cell lines in there, a couple of prostate cancer cell lines in there. The breast cancer cell lines, actually, they were quite sensitive or were relatively sensitive on the more sensitive end, and the prostate cancers were cell lines, at least the ones that we had in our culture, were a little bit more to the higher, the millimolar dose range. But still, they were like that. The highest one was at 5 or 8 millimolar, and which is still in the well below the clinically achievable range. So at least from the preclinical data, there's no reason why they shouldn't benefit. And also the data in the review, there are multiple preclinical studies in breast cancer cell lines and prostate cancer cell lines, also looking at combinations with chemo. Yeah, and then maybe good to add to this. So we did our own experiment in the lab where we tested 51 different cancer cell lines representing different tumor types, and also KRAS mutant and wild types. And basically what was nice to see is that all 51 cell lines were killed in the clinically achievable range. So their IC50 values range from low, from let's say the micromolar sensitivities to low millimolar sensitivities, 5 to 10 millimolar. And when patients get the IV doses, it can go up to 30 millimolar, 15 to 30 millimolar. So basically all the cell lines that we tested were well within this range.
Is there anything to expand upon in terms of what does a patient do if they want to do high dose IVC?
Well, what we see in the literature studies or reported in the scientific literature, I think a lot of it is determined also by practical and financial reasons, probably. So but it's important, the studies that showed efficacy or signs of efficacy and clinical benefit. Also did frequencies of 2 to 3 times a week, and doses that were in the 1.5 gram per kilogram range, so 75 grams at least, 200 grams. And in mono, I think if it's used in monotherapy, then I think 3 times a week would be the minimum for at least the duration of probably 12 weeks or 3 months or so, and then maybe continue maybe a little bit less frequent for another year, or as I would always say, as long as possible and as frequent as possible.
Yeah.
And in the combination treatments, it's oftentimes kind of for logistic reasons. Then people go in when they get their radio or their chemo, and if that's 2 times a week, then the clinical combination study does 2 times a week, because then doing 3 times a week would be logistically too difficult. But at least there are good results obtained in the combination studies where a schedule of 2 times a week was used, but at least it has to be then every week, ideally every week, and then also for at least a duration of 8 weeks. Those were the studies, minimum duration of 8 weeks. Those were the studies that showed signs of benefit.
And were there, was it kind of ultimately associated with a level of vitamin C in your plasma that they were trying to hit and kind of keep a certain level?
Yeah, it's important. So usually it starts with a dose escalation in every patient, and then in which the dose is determined that can reach target plasma values of at least 15 millimolar.
15 millimolar.
And that's usually done in phase 1, and then for phase 2 a dose is picked, and in practice this is between 75 to 90 grams. It looks like every center kind of rediscovers this. And I think every patient, and I think it's a good idea to, in every patient, to do this escalation, because every person is different. But a good estimation can be made from the body weight calculation. But it's, I think usually it's, I'm not a clinician, but the build-up is usually done in a few steps, from like 25 grams and then to 50 and up to usually 1.5 grams. With 1.5 grams per kilogram, usually the target range is reached.
Okay. So you touched on something that was interesting, which is sensitivity. So what you're saying, if I understand you correctly, is that some cancer cell lines just might be more sensitive, meaning they react at lower levels than other cancer cell lines. So while there might be benefit, it might be a range of dose levels that you need to take in order to hit that efficacy amount. Is that true?
Yeah, some are more sensitive than others, but when patients are treated in the clinic, we don't know who is who, and I think we don't need to know. I think in the clinic we just do the better safe than sorry approach, and escalate up to 15 to 30 millimolar plasma levels, and basically with those levels, basically all the cell lines will be killed. Or sorry, cell lines. I mean, potentially it can kill the tumor cells. I think it's the levels can be maintained for 1 or 2 hours, and also we mimicked in our cell line experiments. We mimic this because we only give the drug or vitamin C, ascorbic acid, we pulsed it 2 hours, and then we take it away. We wash the cells and we replace the medium. So that kind of mimics the situation in the clinic, where the peak levels are there for an hour or so. And it depends a bit on the way it's done. Ideally, it's maintained as long as possible. In some studies, it was maintained for 2 or 3 hours at this high peak level, and then it falls off again. And based on that, you can understand that more frequent dosing is always better.
I see. So even with IVC, every day is better than every
Daily
other
is better. The Linus Pauling, the early day Linus Pauling study did daily.
Okay.
It was actually a much lower dose, but it was done daily.
Mm-hmm. Okay. So if you had to, if you could make any decision you want to, you would tell a prospective user of IVC that they might want to go do this daily. But we have seen that every other day, so like 3 days a week, for example,
three days a week is a nice compromise, I think. And if you let's say if your partner can do the IVs, for example, maybe it would be nice to the first 4-6 weeks to have it daily, if that is feasible. Especially if you do it as mono treatment. In a combination treatment, I think it can be less frequent. But basically, the message is the more the better.
In a mono treatment, you mean the only treatment they'd be getting is the IVC versus a layered approach, which would be standard care chemotherapy drugs, for example, plus IVC, correct?
Yes.
Yeah. Okay.
And the mono treatment, I think, would be probably beneficial, especially for people who are so weak that they cannot get the chemotherapy. And I know from my colleagues that in some, or yeah, that there is, I think, in non-small cell lung cancer, for example, people with metastatic disease, I think there's a significant proportion that is actually not fit enough to get this combination chemo.
I see. So it could be.
And some people and some patients, they don't want it. That also happens.
Yeah.
But and then you need to only when the oncologists or the clinicians, they are bound to their protocols, and only when people are really out of options, then more things are possible. But you need to find a physician who can do it and is willing to do it.
Yeah. Okay. Now you mentioned and you touched on safety. What do we need to know about safety? Is this safe for everyone? Do we need to do any testing prior to embarking on a high dose vitamin C regimen to make sure that our bodies will tolerate it well? You mentioned kind of escalating over a bit of time just to make sure that that we seem to be tolerating it well. What do we need to be thinking about for safety?
Luckily, the clinical trials show it's very safe and tolerable, and also actually, and that's not a surprise, because for many decades, complementary and alternative medicine practitioners have administered high dose ascorbate or vitamin C off label without apparent toxicity. But it is important, and that's what they also do, and also is being done in these clinical trials, that patients are properly screened for normal renal function and absence of glucose 6 phosphate dehydrogenase deficiency. Also, that they don't have iron overload or a lot of kidney stones.
So they do screen you a little bit. You mentioned a deficiency or a glucose 6 phosphate. Could you say that?
Glucose 6 phosphate dehydrogenase deficiency that
Yes.
can lead to hemolysis or some problems.
Is that rare? I've never heard of that.
It is rare, but it's good to know this, to check
Okay.
this.
And you can test for it.
Yes.
Could test
Yeah.
for it. So you just
Yeah, they can
ask for
easily,
this test.
in the clinic, they test for that, yes.
Okay. So otherwise, we find it to be very safe. I have to say, in all the studies that I've read, that's been one of the universal findings, is that their patients, not only is this something that is very well tolerated, but that it had, ancillary benefits. So besides killing cancer cells, you had all these other positive benefits that you felt better. And how often is it that a anti-cancer strategy also makes you feel better? That's to
Yeah,
me, that was a big win-win. That does not happen very frequently. Um,
exactly.
so that I thought was pretty awesome. There has been some work suggesting that high levels of antioxidants may not be good for cancer patients, versus if you don't have cancer and you're just trying to prevent ever having to go on this journey, which is terrific. But if you do have cancer, um, is there any danger with ingesting high levels of, so orally taking vitamin C that's working as a antioxidant versus the high dose vitamin C, which is working as a pro-oxidant? Um, should we be afraid of that?
Well, fear that high levels of antioxidant might reduce effectiveness of chemotherapy and radiation for cancer patients turns out to be unwarranted if you look at the practice. Studies that have been performed over many decades show that antioxidants orally used don't harm cancer patients, even when given in conjunction with chemotherapy. And instead, it seems to be more the opposite, that there is a strong positive association of antioxidant use with increased survival. So again, I want to reiterate, patients with cancer are generally very deficient in vitamin C, with detrimental consequences for their quality of life and immune system function. Vitamin C deficiency can result in a long list of symptoms because it's really needed by every cell in the body. And we humans have this genetic defect that predisposes us for hyposcolemia.
Yeah, so just to recap that, because I still can't believe that I had never heard this before I started speaking with you, is that cancer patients' bodies are under a great deal of stress, not only from the tumor and the tumor stress that that's putting on us physically, but also the mental stress of being given a diagnosis of cancer, right? So the stress levels for cancer patients are usually very high. And because we cannot make our own vitamin C, that stress level is sucking all the vitamin C that we have, normal levels, right? Normal levels of eating. It's sucking all of that, and it still needs more because we're in such a heightened sense of heightened state of stress.
Yes, that's
Yeah,
correct. Yeah,
that's
so it's very important for patients with cancer to realize this.
it is. I had never heard of it, so I really appreciate that
But
learning.
I think for every person living uh, a busy life in a modern society. Yeah,
Yeah, that's true. Right. It's not just cancer patients, but we see it exacerbated in
yeah.
cancer patients. So, before we close our interview, I would love to ask you about the future. What do you see as the next steps for cancer patients actively looking for new strategies to help them outperform cancer?
Well, thanks for asking this question. So my lab works on a mass spectrometry based strategy to individualize therapy selection based on an individual patient's tumor profile, the proteome and the phosphoproteome. And we also built a data analysis pipeline that can reveal which kinases are active in the tumor head, and kinases drive a lot of the oncogenic signaling. And from the proteome, we can get other biology and cancer hallmark activity, also which immune subsets are in the tumor. And in the past years, we've worked very hard to basically to get the pipeline going and to do all kinds of preclinical studies to show the value of this approach, what we call like a single sample approach. to basically predict based on a tumor proteome and phosphoproteome what kind of kinase inhibitors could be beneficial for this particular cancer cell or tumor. And we've shown this in colorectal cancer, pancreatic cancer, leukemias, and this has been published in recent years. Now we're also doing retrospective studies where we analyze tumor needle biopsies that were obtained in clinical trials and we generated the profiles, and then we correlate to the patient response, and we can at pre-treatment already let's say predict response or resistance. So altogether, I'm very excited by this research. I mean, these are exciting times for us because I'm around a little bit longer. So I've been doing all my whole scientific life since the early 90s. I've been doing, or mid-90s, I should say, when I started my PhD, working on mass spectrometry, and in the beginning, but to answer biological questions, and now we're answering more biomedical or clinical questions using mass spectrometry. And the field in the past 30 years evolved so much. The machines are super fast, super sensitive. We now have all the bioinformatic tools. We have the genome sequence that totally catalyzed also the whole proteomics field. So I really hope I can basically in the coming maybe 10 years or so that I have probably in my active research life. To translate these findings to the clinic. And my dream is to do a proteome-based clinical trials, and together with the clinicians, to really in a randomized study show the benefit of this approach versus standard of care. But of course, before we can do this, we need to get the funding to do this also. Yeah. And the only thing to add is that I also would be especially interested in combining vitamin C with immunotherapy and with metabolic approaches that patients can do themselves, like fasting mimicking diet.
I encourage all of the patients out there to continue to plug into the work that is being done in this field, and to look at opportunities to fundraise and support the research community, because this is the future. In the meantime, I'm encouraging patients everywhere to layer on strategies to try to improve standard care with additional ideas and therapies, keeping our bodies healthy, and perhaps IVC is going to be right for you to consider, and maybe then it'll help extend your outcomes in a positive direction while we all wait for the big cure that we're all hoping for in the future. So thank you very much for coming. I appreciate your time.
Thank you, Mary Beth, for this opportunity. I should emphasize that I'm a biologist. I'm not a medical doctor, so everything we're discussing here is for educational purposes. Thank you very much. It was my pleasure.
And now, an important announcement. All the information that was provided today was for educational purposes only. I'm a patient, just like you. I am not a licensed or accredited physician, therapist, or clinical researcher. All information provided is not intended as a substitute for the advice of a physician, therapist, nutritionist, or any other qualified healthcare professional.